Here's a statistic that will sound impossible until you think about it. Children born in the 2020s may become the last generation whose biological sex is fixed for life. Not because of social change or cultural shift, but because of biotechnology advancing so rapidly that gender fluidity will soon be written into our cells, not just our identities. Within 30 years, the concept of permanent biological sex may seem as outdated as permanent location seemed before the airplane. Before we start, a quick roll call. Write in the comments which part of this planet you're watching from, and then just listen and enjoy. So here's the question that drives this entire video. How do hormone therapy and genetic switches transform gender from biological destiny into reversible choice? And why does fixed biological sex become optional rather than inevitable? By the end of this journey, you'll understand the precise technologies making this possible, from today's hormone treatments to tomorrow's genetic modifications, the timeline of when these changes arrive, and what happens to human identity when our bodies become as fluid as our minds. You'll see how in vitro gametogenesis lets any cell become sperm or egg, how 3D bioprinted organs give people reproductive systems that match their identity, and how epigenetic editing switches genes on and off without changing DNA itself. This isn't science fiction projected centuries ahead. The research exists now. The technologies are in development. Some are already working in mice, with human trials beginning. Scientists have analyzed DNA from hundreds of transgender individuals and found genetic variations in multiple genes that process sex hormones differently, suggesting biological substrates for gender identity that we're only beginning to decode. Research published recently showed fertility can be recovered in transgender women after stopping hormone therapy, proving more reversibility than previously understood. Teams worldwide have created functional mouse sperm and eggs from skin cells, then use them to produce living offspring. What makes this moment unique in human history isn't just that we understand gender as spectrum rather than binary. Biologists have been saying that for years, identifying intermediate cases that make clean male-female divisions scientifically problematic. What's different now is that biotechnology gives us tools to act on that understanding at the cellular level. Gender reassignment won't mean surgery and lifelong hormone treatments. It will mean activating dormant genetic programs your cells already contain. Think about what permanence meant for every human born before now. Your chromosomes were set at conception. Your hormones organized your brain development before birth, and your body expressed one sexual phenotype that would remain essentially unchanged for decades. You could modify the surface through hormones and surgery, but the deep architecture stayed fixed. That's about to change completely. The fundamental question isn't whether this technology will arrive. It's already arriving. The question is what we do with it once it's here, how we distribute access to it, and how we think about bodies and identity when the biological foundations we've taken for granted for millennia suddenly become negotiable. Here's what that future looks like in practical terms. A person assigned male at birth takes epigenetic therapy that turns off SRY gene expression and upregulates genes that promote female development. Their body produces estrogen naturally without external hormones. Over months, secondary sex characteristics change: fat redistribution, breast development, muscle pattern shifts. But unlike current treatments, this operates at the gene expression level, not through daily pharmaceutical intervention. If they later choose to reverse, a different epigenetic therapy reactivates male pattern genes. The body shifts again, following new genetic instructions. Or consider in vitro gametogenesis, where skin cells transform into induced pluripotent stem cells, then differentiate into primordial germ cells, and finally mature into functional sperm or eggs. This technology, which produced living mouse pups and achieved human proof of concept with skin-to-gamete conversion, means biological males can produce eggs and biological females can produce sperm. Reproduction no longer requires the reproductive anatomy you were born with. A transgender woman keeps her genetic contribution to children without preserving sperm before transition. She simply grows new eggs from her own cells later. Three-dimensional bioprinting adds another dimension entirely. Researchers have successfully transplanted bioprinted ovarian tissue into mice, restoring both fertility and hormone production. Human trials of decellularized tissue matrices have produced live births. The timeline suggests functional 3D bioprinted reproductive organs could be available for transplantation by the 2040s. That means not just functional gametes, but complete reproductive systems. Bioprinted uteruses, ovaries, testes, entire anatomical complexes grown from a person's own cells and implanted where nature put different structures. We're not talking about distant theoretical futures. We're describing technologies already working in animal models, with human applications progressing through research pipelines right now. The scientists doing this work publish in mainstream journals, receive institutional funding, and collaborate internationally. This isn't fringe speculation. It's the leading edge of reproductive biotechnology meeting gender medicine, meeting regenerative biology. And that brings us to the real question. This video explores what happens to gender, to identity, to society itself when biological sex becomes as changeable as hair color. When the boundaries between male and female blur not just socially, but cellularly, when permanence itself becomes a choice rather than a given. Current gender-affirming treatments already show us glimpses of this future. Hormone replacement therapy changes voice pitch, body composition, even some aspects of emotional processing and sensory experience. People who transition describe feeling like they're finally inhabiting their own bodies, like coming home after a long exile. But they also describe the labor of it. Daily medications, monitoring blood levels, managing side effects, navigating healthcare systems that often misunderstand or obstruct their needs. The next generation of technologies eliminates much of that labor. Instead of taking estrogen pills every day, you receive gene therapy that tells your cells to produce estrogen. Instead of testosterone injections every week, you activate the genetic pathways that generate it naturally. Instead of suppressing one set of characteristics while artificially inducing another, you reprogram which genes are active and which are dormant. This isn't just more convenient, it's fundamentally different. Current treatments work against your biology. Future treatments work with it, redirecting existing cellular machinery rather than overwhelming it with external chemicals. The difference is like the difference between painting a wall and replacing the wall entirely. One is surface level, temporary, requiring constant maintenance. The other is structural, integrated, self-sustaining. But here's where it gets really interesting. Once we have the technology to switch genetic expression patterns, once we can turn male development pathways into female ones and vice versa, once we can generate any gamete type from any cell, the categories themselves start to dissolve. Why choose male or female when you can choose specific traits from each? Why accept a package deal when you can customize? Some people will want complete transition from one side of the binary to the other. Others will want something in between, testosterone-driven muscle development, but estrogen-driven fat distribution, or vice versa. Still others will want to shift back and forth, spending years expressing one phenotype, then switching to another. And some will reject the entire framework, creating bodies that don't map to male or female at all. The technology doesn't enforce any particular outcome. It simply opens possibility space that was previously closed. What we choose to do with that space depends on us, our cultures, our values, our visions of human flourishing. And that's the real transformation here. Not just that we can change our bodies in new ways, but that biological sex stops being destiny and becomes design. Stops being something that happens to us and becomes something we decide. The last generation born with permanent gender isn't the last generation of humans. They're the last generation born before we gained full control over this particular aspect of our biology. What comes next is up to us. Welcome to the future, where sex is no longer fate, but choice. Let's start with what exists right now, not decades from now. Walk into a gender clinic today, and you'll receive hormones in pill form, patches, injections, or implants. Transgender women take estrogen and anti-androgens that block testosterone. Transgender men take testosterone that suppresses estrogen production. These are powerful medications that reshape bodies in profound ways, but they work through brute chemistry, flooding the system with external hormones to overwhelm the body's natural production. The process looks straightforward on paper. Estrogen therapy for trans women involves daily pills or weekly patches that deliver a steady stream of female sex hormones into the bloodstream. Add spironolactone or cyproterone acetate to block testosterone receptors, and the body begins shifting within months. Breast tissue develops. Fat redistributes from abdomen to hips and thighs. Muscle mass decreases. Skin softens. Body hair grows slower and finer. Hemoglobin levels drop to female ranges within 4 months. The testicles shrink to less than half their original size. For trans men, testosterone injections trigger equally dramatic changes. Voice drops within 3 to 6 months and never returns to its original pitch, even if hormones stop. Facial hair sprouts and thickens over years. Muscles develop more easily and bulk up with less effort. Fat moves from hips to belly. The clitoris enlarges, sometimes dramatically. Menstruation stops. Skin gets oilier. Acne flares. Body odor changes almost overnight. Some people experience male pattern baldness, depending on their genetics. These aren't subtle shifts. They're wholesale transformations of the body's appearance and function. Changes so profound that many people describe finally feeling at home in their own skin after years or decades of alienation. The psychological benefits are well documented. Reduced depression, decreased anxiety, alleviation of gender dysphoria, higher self-esteem, greater confidence. For many people, hormone therapy is literally life-saving. But here's what the medical literature shows clearly. These changes happen through constant chemical intervention. Stop taking the hormones, and many effects reverse. Trans women who discontinue estrogen see testosterone production resume, body hair return, muscle mass rebuild. Trans men who stop testosterone see menstruation restart, fat redistribution shift back, muscle mass decrease. Some changes are permanent. Breast tissue in trans women, voice deepening in trans men, facial hair once it's grown. But the underlying endocrine system never truly switches over. The body keeps trying to return to its chromosomally programmed default. That's because current hormone therapy doesn't change what the body is trying to do. It just overpowers it. Your gonads still produce the hormones they were designed to produce. Your genes still express the same patterns they always have. You're essentially running one operating system while forcing the display of another through external software that requires constant updates. The practical implications are significant. Trans women take medication every single day for the rest of their lives if they want to maintain their transition. They monitor estrogen and testosterone levels through regular blood tests to make sure they're in female ranges. Too little estrogen and masculinizing features creep back. Too much, and there's increased risk of blood clots, stroke, liver problems. The balance requires careful medical management. Trans men inject testosterone weekly, bi-weekly, or receive implants every few months. They also need regular monitoring, watching for cardiovascular issues, liver function changes, and cholesterol imbalances. High testosterone can convert to estrogen through aromatization, sometimes requiring additional medications to manage. Some people struggle with emotional volatility as their bodies adjust to new hormone levels, experiencing mood swings that settle over time but can be challenging during the transition period. And then there's fertility. Current hormone therapy significantly impacts reproductive function. Trans women see sperm production decline dramatically, often becoming infertile within months of starting treatment. Trans men stop ovulating when testosterone suppresses their menstrual cycles. For years, the standard advice was that these changes were permanent, that people needed to preserve gametes before starting hormones if they ever wanted biological children. Recent research challenges that assumption. Studies from Amsterdam University Medical Center showed that trans women who stopped hormone therapy for several months could restore some sperm production, with about 60% achieving fertility levels sufficient for reproduction. Trans men who discontinued testosterone saw menstrual cycles return and successful pregnancies occur. The body's reproductive systems are more resilient than previously believed. But this resilience comes with a catch. You have to stop the hormones and accept temporary reversal of your transition. For many people, this is psychologically devastating. Having to pause your affirmed gender, watch your body shift back toward characteristics that cause dysphoria, all to maybe achieve fertility, creates an impossible choice. Do you want children, or do you want to remain yourself? There's another limitation that current hormone therapy can't overcome. You're still working with the anatomical structures you were born with. A trans woman can grow breasts, but not ovaries. A trans man can develop facial hair, but not produce sperm from his own cells. Surgical options exist: vaginoplasty, phalloplasty, breast augmentation, facial feminization. But these are reconstructive procedures using existing tissue, not creation of fully functional reproductive anatomy. So while today's hormone therapy produces remarkable changes in secondary sex characteristics, it operates through continuous pharmaceutical intervention rather than fundamental biological reprogramming. It's effective, it's transformative, it's essential for millions of people, but it's also the first generation of what's possible. It's like early computers: functional, valuable, groundbreaking for their time, but primitive compared to what comes next. The real shift happens when we stop overpowering the body's hormonal systems and start reprogramming them. When we change not what chemicals circulate in the bloodstream, but what genetic instructions the cells themselves follow. When transition becomes not a daily pharmaceutical regimen, but a one-time cellular reset that allows the body to maintain its new state naturally. That technology is closer than most people realize. And when it arrives, it will make current hormone therapy look as outdated as bloodletting looks to modern medicine. Here's something that sounds like science fiction, but is actually basic developmental biology. Every human embryo starts with the potential to become either male or female. For the first 6 weeks after conception, we all follow the same developmental path. No discernible difference exists between XX and XY embryos. The tissues that will eventually become either ovaries or testes are identical bipotential structures called gonadal ridges, waiting for a signal to tell them which way to develop. That signal comes from a single gene called SRY, located on the Y chromosome. When SRY activates around week 7, it triggers a cascade of genetic events that push development toward male. Without SRY, the default pathway runs its course and development proceeds toward female. But here's what makes this fascinating. That's not a one-time permanent decision made by unchangeable biology. It's an active process of gene expression that continues throughout life, maintained by ongoing genetic activity. Scientists discovered this by accident when they found naturally occurring humans who didn't follow the expected pattern. People with XY chromosomes, but mutations in SRY developed as female. People with XX chromosomes, but a copy of SRY that had jumped onto a different chromosome developed as male. And in some cases, people developed with ambiguous or intermediate characteristics, demonstrating that male and female aren't binary endpoints, but positions along a developmental spectrum controlled by gene activity. The key insight came from studying sex reversal in animals and rare human cases. Researchers identified a gene called DMRT1 that actively maintains testicular identity throughout life. When they knocked out DMRT1 in adult male mice, the animal's testes began transforming into ovarian tissue. The Sertoli cells that normally support sperm production started expressing genes characteristic of ovarian granulosa cells. The organ that had been definitively male for the animal's entire life began shifting toward female because the genetic program maintaining maleness was switched off. This revealed something profound. Sexual differentiation isn't a permanent state locked in during development. It's an active process continuously maintained by gene expression. Your body doesn't become male or female and then stay that way through inertia. It actively expresses genes that keep it male or female. Turn those genes off and the tissues can shift toward the opposite sex, even in adults. Other key genes in this network include SOX9, which works downstream of SRY to promote testis development, and WNT4, which promotes ovarian development and actively suppresses male pathways. There's FOXL2, which maintains ovarian identity throughout life and must be continuously expressed to prevent adult ovaries from transforming into testes. And there's MAP3K1, a gene that influences sex determination sensitivity, with certain variants associated with differences in sexual development. Research from 2024 analyzed the genetics of hundreds of transgender individuals and found variations in multiple genes involved in sex hormone processing, including variants in the androgen receptor gene estrogen receptor genes, and genes that regulate hormone synthesis and metabolism. The data suggests that gender identity may have biological underpinnings related to how these hormonal signaling pathways function during critical developmental windows. But it goes deeper than hormones. Studies of epigenetic patterns in people with gender incongruence before any hormone therapy show differential DNA methylation in genes critical for brain neurodevelopment, including genes involved in neuronal migration and differentiation. This suggests that epigenetic modifications, the chemical marks on DNA that control which genes are active without changing the underlying sequence, play a role in gender identity at the level of brain development. And here's where the future technology enters. Epigenetic modifications are reversible. Unlike mutations that permanently change DNA sequence, methylation patterns can be added or removed. Histone modifications that control how tightly DNA is packed can be altered. The chemical switches that determine which genes are on or off can be flipped. Technologies already exist that do this in experimental settings. CRISPR-Cas9 systems use a modified version of the gene editing tool that doesn't cut DNA, but instead brings epigenetic modifying enzymes to specific genes. You can target the SRY gene and add repressive marks that silence it. You can target WNT4 and add activating marks that turn it on. You can systematically shift the entire gene expression program from a male pattern to a female pattern or vice versa, without changing a single letter of the genetic code. This has enormous implications. Current hormone therapy works by overwhelming the body's endocrine system with external chemicals. But the gonads keep producing their original hormones. The genetic programs keep trying to express their chromosomally determined patterns, and you need constant medication to maintain the override. Epigenetic therapy would work differently. Reprogram which genes are active. Let the body's own cells produce the hormones naturally. Allow the tissue differentiation pathways to follow new instructions. Imagine a trans woman receiving epigenetic therapy that silences SRY and male pathway genes while activating female pathway genes. Over time, the Leydig cells in the testes that normally produce testosterone could be reprogrammed to produce estrogen instead. The tissue architecture might shift. Eventually, you're not taking external hormones to override your biology. Your biology itself has been reprogrammed to function differently. Or consider a trans man receiving therapy that activates SOX9 and suppresses WNT4 and FOXL2. The ovarian tissue could begin expressing testicular markers. Hormone production could shift from estrogen dominant to androgen dominant. The body generates its own testosterone naturally without external supplementation. This isn't happening in humans yet. The technology exists in laboratory settings, proven in cell cultures and animal models, but applying it safely to living people requires years of additional research. The questions are massive. How do you target epigenetic modifications to the right tissues without affecting others? How do you ensure the changes persist long-term? What are the unintended consequences of reprogramming sexual differentiation pathways in adults whose bodies developed according to different programs for decades? But the principle is established. Sex differentiation is controlled by active gene expression. Those expression patterns can be modified through epigenetic mechanisms, and the technologies to make those modifications exist and are rapidly improving. Within a generation, the ability to reprogram sexual biology at the genetic level may move from theoretical possibility to clinical reality. And when that happens, transition stops being a matter of overpowering your biology with external chemicals and becomes a matter of rewriting your biology at its source. The implications of that shift are what we explore next. Let's talk about what makes you biologically male or female at the most fundamental level. It's not actually your chromosomes, though that's what everyone learns in school. Having XY chromosomes doesn't automatically make you male, and having XX doesn't automatically make you female. What matters is which genes are turned on and which are turned off, which proteins get made and which don't, which cellular pathways activate and which remain dormant. This is the domain of epigenetics, the layer of biological control that sits on top of your DNA sequence and determines what your genes actually do. Think of your genome as a massive library containing instructions for building and running a human body. Epigenetic marks are like bookmarks and sticky notes that tell your cells which books to read and which to ignore. The books themselves don't change, but which ones get read makes all the difference between a liver cell and a brain cell, between male development and female development. The key epigenetic mechanisms are DNA methylation, where chemical groups attach to DNA and typically silence genes, and histone modification, where proteins that package DNA get chemically tagged to make genes more or less accessible. These marks can be added, removed, and changed throughout life. They respond to hormones, environmental signals, even experiences and behaviors. They're dynamic, flexible, reversible. And here's what makes this revolutionary for gender transition. Epigenetic patterns differ systematically between males and females, particularly in genes involved in sexual differentiation and hormone response. Research has identified specific methylation differences in transgender individuals compared to cisgender individuals, even before any hormone therapy begins. These differences appear in genes critical for brain development, neuronal function, and hormone receptor activity. Studies of brain tissue show sex-specific epigenetic signatures that develop early in life and persist into adulthood. Certain genes in the hypothalamus, the brain region involved in gender identity, show different methylation patterns in males versus females. When researchers examined brain tissue from transgender individuals, they found epigenetic patterns that sometimes aligned more closely with their identified gender than their assigned sex. This suggests something profound. Gender identity may be partially encoded in epigenetic marks laid down during fetal development or early childhood. Marks that create a mismatch when they don't align with chromosomal sex and gonadal hormones. If that's true, then gender dysphoria isn't just psychological or purely social. It has a biological substrate in the epigenetic programming of the brain. But here's where it gets really interesting. If gender-related traits are controlled by epigenetic marks, and epigenetic marks are reversible, then theoretically, we could reprogram them. We could change the pattern of gene expression throughout the body without touching the underlying DNA sequence. The technology to do this already exists in experimental form. It's called epigenetic editing, and it uses modified versions of CRISPR, the gene editing system that won the Nobel Prize. Traditional CRISPR cuts DNA to make permanent changes. Epigenetic CRISPR uses a deactivated version called dCas9 that can't cut, but can still find specific genes. Instead of cutting, it brings along enzymes that add or remove epigenetic marks. Want to silence the SRY gene that drives male development? Use dCas9 to bring methylation enzymes to the SRY promoter, adding chemical marks that shut it down. Want to activate genes like WNT4 that promote female development. Bring acetylation enzymes that open up the DNA and make those genes accessible. You can target multiple genes simultaneously, creating a coordinated shift in expression patterns across entire developmental pathways. This has been demonstrated in cell cultures and animal models. Researchers have used epigenetic editing to control gene expression with precision, turning genes on or off for sustained periods without changing the DNA sequence. The effects can last weeks, months, potentially longer if the epigenetic machinery maintains the marks through cell division. Now imagine applying this to gender transition. Instead of taking hormones daily to override your body's natural production, you receive epigenetic therapy that reprograms your cells to produce different hormones naturally. The therapy targets hormone-producing cells in the gonads, changing their epigenetic state so they express genes for estrogen synthesis instead of testosterone, or vice versa. Your body would generate its own hormones at appropriate physiological levels, regulated by the same feedback mechanisms that normally control hormone production. No daily pills, no injections, no external chemicals. Just your own cells following new instructions about which genes to express. But it goes beyond hormones. Epigenetic therapy could reprogram other tissues too. Adipose tissue that normally follows male distribution patterns could be reprogrammed to follow female patterns. Sebaceous glands that produce male pattern skin oils could shift to female patterns. Hair follicles programmed for male pattern growth could change their programming. The result would be transition at the cellular level, where individual cells throughout the body shift their gene expression profiles from one sex-typical pattern to another. It's not cosmetic change or pharmaceutical override. It's fundamental biological reprogramming. And crucially, it could be reversible. Current hormone therapy is partially reversible if you stop taking medications, but many changes are permanent. Epigenetic therapy could be fully reversible in principle. If you receive treatment that methylates male pattern genes and demethylates female pattern genes, you could later receive the opposite treatment that removes those marks and restores the original pattern, or creates a new pattern entirely. This opens possibilities that current medicine can't touch. You could transition from male to female, live that way for years or decades, then transition back if you chose, or transition to an intermediate state that expresses some male traits and some female traits simultaneously. The binary disappears when you have control at the epigenetic level. Research published recently examined how current hormone therapy affects epigenetic patterns in transgender individuals. The study found that hormone treatment does create some epigenetic changes, shifting methylation patterns in predictable directions. But these changes are secondary effects of altered hormone levels, not primary reprogramming of cellular identity. True epigenetic therapy would work the other way around. Reprogram the cellular identity first. Let hormone levels shift as a consequence of that reprogramming, and allow all the downstream changes to follow naturally from the new gene expression program. The challenges are significant. Delivering epigenetic editing tools to the right cells throughout the body is technically difficult. Ensuring the changes persist long-term requires either continuous expression of the editing machinery or creation of self-maintaining epigenetic states. Understanding which genes to target and which marks to place requires mapping the complete epigenetic landscape of sexual differentiation. Safety is paramount. Epigenetic changes affect many cellular processes beyond sexual characteristics. Unintended off-target effects could disrupt other gene expression programs. Long-term consequences of reprogramming adult cells that developed under different epigenetic instructions are unknown. But the principle is sound. The technology exists in early forms, and research is progressing rapidly. Within a decade or two, epigenetic transition therapy could move from theoretical possibility to experimental trials to clinical reality. When it does, it will transform what transition means, making it not a pharmaceutical workaround, but a biological reset at the most fundamental level of gene regulation. Here's a fact that sounds impossible. Within the next decade, scientists will likely be able to take a small sample of your skin, reprogram those cells in a laboratory, and transform them into functional sperm or eggs capable of creating a living child. Not science fiction, not distant speculation. Actual biotechnology already working in mice and advancing rapidly toward human application. This process is called in vitro gametogenesis, abbreviated IVG, and it represents perhaps the most radical transformation of human reproduction since the invention of in vitro fertilization 50 years ago. IVF took eggs and sperm that already existed and combined them outside the body. IVG creates new eggs and sperm from cells that were never reproductive cells at all. The implications for gender and biological sex are staggering. A biological male could produce eggs. A biological female could produce sperm. Reproduction would no longer require the reproductive anatomy you were born with. Two people of the same biological sex could have children that are genetically related to both of them. A single person could produce both eggs and sperm and, theoretically, reproduce alone. Let's break down how this actually works, because the science is as elegant as it is revolutionary. Every cell in your body contains the same DNA, the complete genetic instruction set for building a human. A skin cell and a sperm cell have identical genomes. What makes them different is which genes are active and which are silenced, which cellular programs are running and which are shut down. Scientists discovered about 15 years ago that you can take a fully differentiated adult cell, like a skin cell, and reprogram it back to a pluripotent state, essentially rewinding it to an embryonic-like condition where it can become any cell type. These are called induced pluripotent stem cells, or iPSCs. The researcher who discovered this, Shinya Yamanaka, won the Nobel Prize for it because the implications touch virtually every field of medicine. Once you have iPSCs, you can coax them to differentiate into other cell types by providing the right chemical signals and growth factors. Want heart muscle cells? Use these signals. Want neurons? Use those signals. Want primordial germ cells, the precursors to sperm and eggs? That's where IVG comes in. Researchers first achieved this in mice. They took skin cells, created iPSCs, then differentiated those iPSCs into primordial germ cells. They implanted those cells into mouse testes or ovaries, where the cells completed their development into mature sperm or eggs. They used those lab-created gametes for in vitro fertilization. And they got living, healthy mice that grew to adulthood and could reproduce normally themselves. That was proof of concept. The next step was making the entire process work in vitro, outside the body, without needing to implant cells into living gonads. In 2023, researchers succeeded in creating functional mouse eggs entirely in laboratory culture, from iPSCs to mature eggs capable of fertilization, all in a dish. They fertilized those eggs and produced living mouse pups. Then came the advances with human cells. In 2024, a team successfully converted human skin cells into cells resembling very early primordial germ cells, the first stage of gamete development. They couldn't complete the full process to mature eggs or sperm yet, but they demonstrated that human cells could be pushed onto the germ cell developmental pathway. The technical challenges are immense. Germ cells are incredibly complex, with unique genetic features not found in other cell types. They undergo meiosis, the special cell division that halves the chromosome number, so that when sperm meets egg, the resulting embryo has the correct number. Getting that process to work correctly in laboratory conditions requires precise control of cellular environment and developmental timing. There are also epigenetic considerations. Germ cells undergo extensive epigenetic reprogramming, erasing and resetting the chemical marks on DNA that control gene expression. This is crucial because these marks get inherited by offspring. Errors in epigenetic reprogramming could cause developmental problems or disease susceptibility in children conceived through IVG. But researchers are making steady progress. Multiple teams worldwide are working on different aspects of the problem, optimizing the conversion of iPSCs to germ cells, developing culture systems that support meiosis, ensuring proper epigenetic reprogramming, validating the safety and function of IVG-derived gametes. Timeline estimates vary. Optimistic researchers suggest functional human IVG could be demonstrated within 5 years. Conservative estimates push that to 10 or 15 years. Regulatory approval and clinical implementation would take additional years beyond initial proof of concept, but the trajectory is clear. This technology is coming. Now consider what this means for transgender reproduction. Currently, transgender people face difficult choices about fertility. Starting hormone therapy usually impairs fertility, sometimes irreversibly. The standard advice is to preserve gametes before transition, sperm banking for trans women, egg freezing for trans men. But that freezes you at your birth sex biologically. A trans woman preserves sperm, so any children would have her genetic contribution as a biological father, not a biological mother. A trans man preserves eggs, contributing genetically as a biological mother, not a biological father. For many people, this creates psychological dissonance, maintaining a biological connection to the sex they're transitioning away from. IVG eliminates that dissonance. A trans woman could transition fully, live as female for years or decades, then use IVG to create eggs from her own cells. Those eggs would carry her genetic contribution as a biological mother. She could have children without any preserved connection to her pre-transition biology. Similarly, a trans man could create sperm from his own cells through IVG, contributing genetically as a biological father. The technology doesn't care what reproductive anatomy you currently have. It only needs cells with your DNA. This gets even more interesting when you combine IVG with the other technologies we've discussed. Imagine someone who has undergone epigenetic therapy to transition from male to female. Their cells now express female pattern genes. When those cells are reprogrammed into iPSCs and then into germ cells, do those germ cells follow male or female developmental pathways? Could the epigenetic reprogramming during IVG be deliberately guided to produce eggs instead of sperm, regardless of whether the person has XX or XY chromosomes. Some of this is speculative, pushing beyond what current research has demonstrated. But the basic principle holds. If you can create germ cells from any cells, and if sexual differentiation is controlled by gene expression rather than chromosome identity alone, then theoretically, any person could produce either type of gamete. For same-sex couples, this opens unprecedented reproductive possibilities. Two women could both contribute genetically to a child. One provides the egg naturally, the other uses IVG to create sperm. Two men could both contribute. One provides sperm naturally, the other uses IVG to create an egg. The child would be biologically related to both parents in the traditional genetic sense. There are concerns, of course. Children born from IVG-created gametes, where both parents are the same chromosomal sex, would have unusual genetic configurations. Two biological females would produce a child with two X chromosomes, always female. Two biological males would produce children that are either XY, male, or YY, non-viable. The sex ratio and genetic diversity implications need careful consideration. There are ethical questions about consent and oversight. Should IVG be restricted to treating infertility, or should it be available for anyone who wants it? What regulations should govern its use? How do we ensure safety when the long-term effects on offspring won't be known for generations? And there are profound social implications. When reproduction decouples completely from sexual anatomy, when anyone can produce any type of gamete, when biological sex becomes irrelevant to reproduction, the very foundation of gender categories starts to erode. If a trans woman can be a biological mother, and a trans man can be a biological father, what do those categories even mean anymore? This is the world IVG is creating, one where reproductive biology is as flexible as gender identity, where having children doesn't require having the body parts traditionally associated with your role in conception, where the biological contribution you make to the next generation can match your affirmed gender rather than your assigned sex. It's not here yet, but it's coming. And when it arrives, it will complete another piece of the transition from permanent, fixed biological sex to fluid, chosen gender, expressed at every level, from social identity to cellular function to genetic contribution to future generations. Imagine walking into a clinic, not for surgery, but for construction. Not reconstruction of what was damaged, but construction of what was never there. A transgender woman receiving a bioprinted uterus grown from her own cells. A transgender man receiving bioprinted testes that produce testosterone naturally. Complete reproductive organs, fully functional, built to specification in a laboratory and transplanted into bodies that nature assembled differently. This isn't distant fantasy. The foundational technologies exist right now. Researchers have already bioprinted organs, transplanted them into living animals, and demonstrated function. We're not talking about prosthetics or mechanical devices. We're talking about living tissue, grown cell by cell, vascularized and innervated, capable of integrating with the body's existing biology. Three-dimensional bioprinting works by depositing living cells layer by layer, following a digital blueprint, building up three-dimensional structures. Think of it as a biological version of 3D printing. But instead of plastic filament, you're using bioink made from living cells suspended in biocompatible materials. The printer head moves precisely through three-dimensional space, placing cells exactly where they need to be to create tissue architecture. The process starts with cells. often stem cells that can differentiate into multiple tissue types. These cells are mixed with hydrogels that provide structural support and contain growth factors that guide cellular behavior. The BioInk is loaded into printer cartridges. The printer deposits thin layers according to a computer model of the target organ, building up the structure gradually. As layers accumulate, the cells begin interacting, forming connections, organizing themselves into functional tissue. Early bioprinting focused on simple tissues, sheets of skin for burn victims, cartilage for joint repair, bladder tissue for reconstruction after cancer surgery. These relatively simple structures prove the concept, but organs are far more complex. They require multiple cell types organized in precise three-dimensional arrangements. They need blood vessels to deliver nutrients and oxygen. They need innervation to connect to the nervous system. They need integration with the body's hormonal and immune systems. Researchers have tackled these challenges systematically. For vascularization, they bioprint channel structures that endothelial cells can line, creating blood vessels throughout the organ. For innervation, they incorporate signals that attract nerve growth. For integration, they use the recipient's own cells to minimize immune rejection. The success stories are remarkable. Scientists have bioprinted ovarian tissue and transplanted it into mice that had their ovaries removed. The bioprinted tissue survived, formed follicles containing eggs, restored hormone production, and enabled the mice to conceive and give birth to healthy pups. The offspring were normal, grew to adulthood, and reproduced themselves. Other teams have worked with decellularization, a related but different technique. They take donor organs, flush out all the original cells, leaving behind just the structural scaffold of extracellular matrix. Then they reseed that scaffold with the recipient's own cells. The scaffold provides the correct three-dimensional architecture, while the recipient's cells provide the actual functional tissue, dramatically reducing immune rejection. This approach has produced successful transplants in humans. Women born without uteruses received lab-grown uterine tissue, achieved pregnancies, and delivered healthy babies. The tissue integrated with their bodies, responded to hormonal signals, supported fetal development through 9 months of pregnancy, and functioned well enough for successful childbirth. Now apply these technologies to transgender medicine. The core principle is the same. Build the organs someone should have but doesn't, using their own cells to minimize rejection, creating fully functional reproductive anatomy that matches their gender identity rather than their assigned sex. For transgender women, that means bioprinting ovarian tissue capable of producing estrogen and progesterone, potentially even containing follicles with eggs created through in vitro gametogenesis. It means bioprinting a uterus with proper muscular walls, endometrial lining, blood supply, and connections to existing anatomy. It means creating a complete female reproductive system in someone born male. The technical challenges are substantial. The human uterus is a complex organ with multiple tissue types: smooth muscle that contracts during labor, endometrium that cycles monthly in response to hormones, cervix with specialized mucus-producing cells, blood vessels that expand dramatically during pregnancy. Getting all these components to function correctly requires precise replication of the organ's architecture and cellular composition. But the progress is steady. Researchers have successfully bioprinted uterine tissue from human cells and demonstrated that it responds appropriately to hormonal signals. They've created miniature organs called organoids that recapitulate key features of full-sized organs in laboratory dishes. They've mapped the cellular composition and three-dimensional structure of reproductive organs in unprecedented detail, using advanced imaging and sequencing technologies. For transgender men, the focus is on testes and prostate. Testicular tissue is extraordinarily complex, with multiple specialized cell types organized in precise structures called seminiferous tubules where sperm develop. Leydig cells between the tubules produce testosterone. Sertoli cells within the tubules support developing sperm. The whole system operates under tight hormonal regulation. Creating functional testicular tissue through bioprinting would mean not just making something that looks like a testis under a microscope, but making tissue that actually produces testosterone at physiological levels, responds to hormonal feedback from the pituitary gland, and potentially even produces sperm through the combination of bioprinting and in vitro gametogenesis. Early experiments are promising. Researchers have bioprinted testicular organoids containing Leydig cells that produce testosterone. They've created structures that recapitulate some features of seminiferous tubules. They've demonstrated that bioprinted testicular cells can survive transplantation and maintain function for weeks to months in animal models. The timeline for human application depends on overcoming several hurdles. Safety is paramount. Bioprinted organs must function reliably for years or decades without causing complications. They must integrate properly with existing anatomy, connect to blood vessels, and respond appropriately to the body's regulatory signals. Long-term studies in animal models are essential before moving to human trials. Regulatory approval will take time. Bioprinted organs represent a new category of medical intervention, not quite transplantation and not quite tissue engineering. Regulatory agencies need to develop frameworks for evaluating their safety and efficacy. Clinical trials must demonstrate not just that the organs can be implanted successfully, but that they provide meaningful benefit without unacceptable risks. Cost is another consideration. Current bioprinting technologies are expensive, requiring specialized equipment, highly trained personnel, and weeks of laboratory work to create a single organ. As the technology matures and scales up, costs should decrease, but initially bioprinted organs will likely be available only at specialized medical centers and covered by insurance only in specific circumstances. Despite these challenges, the trajectory is clear. Multiple research groups worldwide are working on bioprinting reproductive organs. Funding is flowing from government agencies, foundations, and private investors. Publications demonstrate steady progress in solving technical problems and achieving functional milestones. Conservative estimates suggest bioprinted reproductive organs could enter clinical trials for humans within the next decade, with broader clinical availability by the 2040s. When that happens, gender transition will include not just hormones and surgery, but biological construction. Transgender individuals won't just modify their existing anatomy. They'll add completely new organs, grown from their own cells, fully functional, permanently integrated into their bodies. A transgender woman won't just take estrogen pills. She'll have ovaries that produce it naturally. A transgender man won't just inject testosterone. He'll have testes that generate it endogenously. This completes the biological transformation in a way no current technology can. It moves beyond modification toward genuine reconstruction, creating bodies that match gender identity at every level, from gross anatomy to cellular function to hormonal production. Combined with epigenetic editing and in vitro gametogenesis, bioprinted organs represent the final piece of the puzzle, making biological sex not just reversible or modifiable, but fully designable from the ground up. We've talked about the technologies: epigenetic editing that reprograms gene expression, in vitro gametogenesis that creates gametes from any cells, bioprinted organs that construct new anatomy. Now let's talk about what happens when these technologies become widely available. Not just what's possible in laboratories, but what becomes normal in society. Because technology doesn't just change what we can do. It changes how we think, what we value, who we become. Start with a simple scenario. It's 2045. A teenager visits a clinic complaining of gender dysphoria, the distressing sense that their body doesn't match their gender identity. 20 years earlier, they would have received psychological counseling, possibly hormone therapy starting in their late teens, maybe surgery years later. But now, the doctor explains different options: epigenetic therapy that will reprogram their cells to follow the opposite sex's developmental pathway, treatment that takes a few months to fully manifest, but results in their body naturally producing the hormones and expressing the characteristics they identify with. The teenager starts treatment. Within 6 months, their secondary sex characteristics begin shifting. Within a year, the changes are substantial. Within 2 years, the transition is complete. But here's what's different from 2025. They're not taking daily medications. Their own cells are producing the hormones. Their body has been fundamentally reprogrammed at the molecular level. And if they change their mind at any point, the process can be reversed or adjusted with different epigenetic therapy. Now multiply that scenario by millions. Gender transition becomes not a rare, difficult, partially reversible medical intervention, requiring years of commitment and significant risk. It becomes a relatively straightforward, fully reversible, biologically integrated option that people can pursue at any age with reasonable confidence about the outcome. What happens to the concept of transgender versus cisgender in that world? Currently, those categories map onto real differences. Cisgender people's gender identity matches their birth sex. They never undergo transition. Transgender people's identity doesn't match. They transition if they can access treatment. But when transition becomes easy and reversible, when even cisgender people can modify their biology for aesthetic or functional reasons, the boundary blurs. Consider a cisgender woman who wants more muscle mass but doesn't want to lose her female fat distribution or breast tissue. With current technology, taking testosterone would masculinize her completely, which she doesn't want. With epigenetic therapy, she could selectively activate just the genes related to muscle development without affecting other aspects of sexual dimorphism. She's not transitioning to male. She's customizing her biology to express specific traits. Or a cisgender man who wants less body hair and softer skin, but maintains his deep voice and facial structure. Current technology can't offer that level of specificity. Epigenetic and bioprinting technologies potentially could. He modifies his biology without changing his gender identity or social role. These people aren't transgender in the current sense. They identify with their assigned sex, but they're also not leaving their biology unchanged. They're in a third category, people who modify their sex characteristics for reasons other than gender dysphoria. And once that becomes common, the distinction between modifying because you're transgender versus modifying for other reasons starts to dissolve. The deeper transformation is philosophical. Currently, we think of biological sex as something that happens to us, a result of chromosomes and prenatal development that we didn't choose and can't fundamentally change. We can modify the surface through hormones and surgery, but we can't change the underlying reality. Gender identity is what we choose or discover about ourselves. Biological sex is what nature imposes. But when biological sex becomes fully modifiable, when we can reprogram our cells, construct new organs, generate any type of gamete, that entire framework collapses. Biological sex stops being something imposed by nature and becomes something we design. It moves from the category of given to the category of chosen. That's an enormous shift. For all of human history, certain aspects of our bodies were simply facts we had to accept. You were born male or female, and that determined vast swaths of your biology, from hormone levels to reproductive capability to physical appearance. You could be dissatisfied with it. You could identify differently, but you couldn't fundamentally change it. The technologies we're discussing eliminate that constraint. They make biological sex as malleable as hair color or body weight. Something you can change if you want to, as many times as you want to, in whatever direction you want to. Not just male to female or female to male, but potentially to intermediate states or entirely novel configurations. And that raises questions nobody has seriously grappled with because they've never been practically relevant. If you can switch your biological sex back and forth multiple times over your lifetime, what does sex even mean? Is it a stable category or a temporary state? If you spend 20 years male, 30 years female, and 20 years in various intermediate configurations, what is your sex? Or consider relationships and reproduction. Currently, reproductive roles are tied to biological sex in ways that limit relationship configurations. Two biological males can't both contribute genetically to a child through natural reproduction. Two biological females can't either. Those limitations shape how we think about family structures, parenting roles, genetic relationships. When in vitro, gametogenesis makes any person capable of producing either gametes, when bioprinted organs make any person capable of gestation, those limitations vanish. Two people of the same chromosomal sex can both contribute genetically to a child. A single person could produce both egg and sperm and technically reproduce alone. The biological constraints that have always shaped human relationships and family structures simply disappear. What happens to social categories based on sex when the underlying biological reality becomes fluid? We have separate sports leagues, different medical protocols, legal protections based on sex. Those categories make sense when sex is stable and clearly defined. They become much more complicated when sex is fluid and customizable. A person could be biologically male when they compete in sports, then transition to female for other contexts, then back to male. How do we handle that? Do we categorize based on current biology, past biology, genetic sex, or something else entirely? Each option has problems. None maps cleanly onto a world where biological sex is optional and reversible. Or consider medical treatment. Many diseases affect males and females differently. Cardiovascular disease presents differently, cancer risks vary, drug metabolism differs. Those differences are tied to hormone levels, genetic factors, anatomical structures. When all of those become modifiable and people change them over time, how do doctors assess risk and choose treatments? There are equity questions too. These technologies will be expensive initially. Early adopters will be wealthy people in developed countries. That creates scenarios where gender fluidity is a privilege available to some, but not others. Does that exacerbate inequality? Do we have an obligation to make these technologies universally accessible? If so, who pays for it? There are questions about informed consent, especially for young people. If a teenager can receive epigenetic therapy that fundamentally reprograms their biology, and the process is reversible, but reversal requires another intervention, at what age can someone consent to that? Do we require psychological evaluation first? How long a waiting period? The same questions that apply to current transition procedures apply with even more force when the interventions go deeper into biology. And there are profound questions about identity and authenticity. Some people argue that accepting your body as it is, regardless of how you feel about it, is part of being human. That the constraints of biology are part of what gives life meaning. That unlimited modification of the body erodes something essential about personhood. Others argue the opposite, that being able to make your body match your identity is liberation. That removing arbitrary biological constraints enables human flourishing. That authenticity means being able to become who you truly are, rather than accepting what nature randomly assigned you. Both perspectives have merit. Both will shape social debates as these technologies develop. The question isn't which is correct, but how societies navigate between them, balancing individual autonomy with social concerns, medical possibility with ethical limits, innovation with caution. What seems certain is that the next generation will face choices about biological sex that no previous generation has encountered. And those choices will reshape not just individual lives, but the entire social architecture built around sex and gender. We're watching the emergence of a fundamentally different human condition, one where the biology we're born with is a starting point rather than a fixed endpoint, where the body is something we continuously design rather than passively inhabit. That future is neither utopia nor dystopia. It's complex, full of opportunities and challenges, requiring wisdom we're only beginning to develop. But it's coming, faster than most people realize. And how we respond to it will define what it means to be human in the decades ahead. We've traveled through technologies that sound like science fiction, but represent actual scientific trajectories. Epigenetic editing that reprograms gene expression, in vitro gametogenesis that creates gametes from any cells, bioprinted organs that construct new anatomy. Each technology alone is revolutionary. Together, they dismantle the most fundamental biological constraint humans have ever known, the permanence of sex. But let's pause before concluding and sit with the strangeness of what we're describing. For the entire span of human existence, across every culture and every era, biological sex has been destiny. You were born male or female, and that determined enormous swaths of your life. Your body, your reproductive role, your hormonal profile, aspects of your health and mortality. You could be unhappy with it. You could transcend social expectations around it, but you couldn't fundamentally change the biology itself. That constraint shaped everything. It shaped how societies organized themselves, how labor divided, how families formed, how power distributed. It shaped philosophy and religion, art and literature, law and custom. The permanence of sex was one of the bedrock facts of human existence, as unchangeable as mortality itself. And now we're describing technologies that eliminate it, that make sex not a permanent state, but a temporary configuration. Not destiny, but design. Not what nature imposes, but what individuals choose. This isn't a small shift at the margins. It's the removal of a constraint that has defined the human condition since our species emerged. Here's the paradox at the heart of this transformation. When everything becomes possible, permanence itself becomes a choice. The last generation, born with permanent gender, aren't losing something that future generations will have. They're the last to experience something future generations will have to actively choose: a fixed biological sex maintained throughout life. Think about what choice means in that context. Currently, if you're comfortable with your assigned sex, you don't choose it. You just are it. It's the default, the baseline, the state you remain in unless you actively decide to transition, which requires enormous effort and comes with significant limitations. But in a future where biological sex is fully fluid, staying the same requires a choice just as much as changing does. Each year, each decade, you could modify your biology if you wanted to. The technology exists. The option is available. Choosing not to use it is itself a decision, one you implicitly make repeatedly. Permanence stops being what happens automatically and becomes what you actively maintain. Even cisgender identity transforms from passive acceptance to active commitment. That's philosophically profound. It means there's no natural baseline anymore, no default state, no configuration that's simply what you are rather than what you choose to be. Everything becomes intentional. Your body stops being something that happens to you and becomes something you continuously decide. Some people will find that liberating. The ability to design your own biology, to express yourself physically in ways that match your identity and preferences, to change and evolve over time as your sense of self develops. Freedom from biological constraints that feel arbitrary and limiting. The chance to be who you truly are rather than accepting what random genetics assigned you. Others will find it destabilizing. If nothing about your body is given, if everything is chosen, where's the ground to stand on? What does authenticity mean when you can modify yourself at will? Is there still a you underneath all the modifications? Or does the self become just a series of design choices? Some people might experience the weight of constant choice as burden rather than liberation. These aren't questions with clear answers. They're tensions we'll have to navigate as these technologies develop. Both perspectives have validity. Both capture something real about human experience. The challenge is creating space for multiple responses, allowing some people to embrace modification while others maintain permanence, without either choice becoming mandatory or stigmatized. There are ethical questions we haven't fully explored. Should there be age limits for biological sex modification? If a child expresses gender dysphoria at age 8, do we provide epigenetic therapy immediately, or wait until they're older to ensure the decision is truly theirs? Current debates about puberty blockers and adolescent transition will intensify dramatically when the technologies become more powerful and the changes more fundamental. What about prenatal modification? If we can detect gender identity markers in utero, or modify the fetus to align with expected preferences, should we? That touches on deeply controversial territory around designer babies and eugenics. Most people would probably object to parents choosing their child's gender. But what if the child has markers suggesting they'll experience gender dysphoria? Is prevention different from selection? Then there are questions about access and inequality. Cutting-edge medical technologies are always expensive initially. Early adopters of biological sex modification will likely be wealthy people in developed countries. That creates a world where gender fluidity is available to some, but not others, where your ability to align your body with your identity depends on your economic class. Is that acceptable? Do we have an obligation to make these technologies universally accessible? How do we balance innovation incentives with equity concerns? There are also questions about social institutions. We organize much of society around biological sex. Sports leagues, medical protocols, legal categories, bathrooms and changing rooms, data collection systems. Those organizational schemes make sense when sex is stable and binary. They become vastly more complicated when sex is fluid and customizable. Societies will need to develop new frameworks that accommodate biological diversity without completely abandoning categories that serve useful purposes. And there are questions about meaning and value. Does something precious get lost when biological constraints disappear? Some philosophical and religious traditions argue that accepting our given nature, working within constraints, finding meaning despite limitations, is essential to human dignity and spiritual growth. That unlimited self-modification is a form of hubris, an attempt to escape the human condition rather than embrace it. Others argue that transcending biological constraints is exactly what humans do, that we've always modified ourselves and our environment, that using technology to align our bodies with our identities is no different in principle from using technology to cure disease or extend life, that the human condition is characterized by our ability to change our circumstances, not by passive acceptance of them. Both views will shape how societies approach these technologies. Neither is obviously wrong. The wisdom is in recognizing the validity of both perspectives while finding practical ways to navigate between them. What seems certain is that we're entering a period of profound transformation in how humans relate to their own biology. For the first time, we're gaining the ability to redesign fundamental aspects of ourselves that have always been fixed. That power comes with immense opportunities and serious responsibilities. The opportunities are clear. People suffering from gender dysphoria could align their biology with their identity fully and reversibly. People could customize their bodies to express themselves in new ways. Reproductive possibilities could expand dramatically. Human diversity could flourish in forms we can barely imagine. The responsibilities are equally clear. We need to ensure access is equitable, that individuals have true autonomy in their choices, that we don't create new forms of coercion or pressure, that we think carefully about long-term consequences for individuals and societies, that we maintain space for different values and perspectives. We're not at the end of this story. We're at the beginning. The technologies are emerging, but not yet mature. The social transformations are starting, but far from complete. The ethical frameworks are developing, but not yet adequate to the challenges ahead. What we can say is this: children born today may indeed be the last generation for whom biological sex is permanent and fixed. Their children and grandchildren will live in a world where sex is fluid, customizable, reversible, where the body is a canvas for ongoing expression rather than a fixed fact, where human diversity expands beyond anything previous generations imagined possible. That world isn't inherently good or bad. It's different, profoundly so, in ways that create both opportunities and challenges. How we navigate that difference, how we balance freedom and wisdom, innovation and caution, diversity and coherence will define what it means to be human in the centuries ahead. The future isn't determined. It's something we create through our choices, our values, our willingness to grapple honestly with difficult questions. The technologies we've discussed will arrive. What we do with them, how we integrate them into human life, what kind of society we build around them, remains to be seen. One thing is certain: we're witnessing the emergence of a fundamentally new human condition. Not better or worse necessarily, but different in ways that challenge everything we thought we knew about sex, gender, identity, and what's possible for human beings. That's simultaneously exhilarating and sobering, full of possibility and full of responsibility. Welcome to the future, where biological sex is no longer fate, but choice, where permanence itself becomes optional, where the last generation born with fixed gender gives way to generations who continuously design themselves. It's here, it's happening. And what comes next depends on us. We've covered the science, the genetic switches, the epigenetic reprogramming, the bioprinted organs, the gametes created from skin cells. We've explored the social transformations, the blurring of transgender and cisgender categories, the dissolution of sex-based social structures, the emergence of biological customization as normal rather than exceptional. But there's a dimension we haven't fully explored: the deeply personal human experience of living through this transition. Because that's what we're really talking about. Not just technological possibility or social change, but a fundamental shift in what it means to inhabit a body, to have a gender, to reproduce, to understand yourself as male or female or something else entirely. These aren't abstract philosophical questions. They're intensely personal experiences that will shape millions of lives in the coming decades. Let's start with something simple: the experience of choice. Right now, if you're cisgender, comfortable with your assigned sex, you don't think much about your body's sex characteristics. They're just there, part of the background of existence, like breathing or having a heartbeat. You don't choose to be male or female. You just are. But when biological sex becomes fully modifiable, that passive acceptance transforms into active decision. Each morning, you wake up in a body you could change if you wanted to. The technology exists. The option is available. Staying the same becomes a choice you make repeatedly throughout your life. For some people, that will feel empowering. They actively choose their body, affirming daily that this is who they want to be. Their gender identity becomes something they own fully, not just psychologically, but biologically. There's a satisfaction in that, a sense of agency and self-determination that previous generations never experienced. For others, it might feel burdensome. The weight of constant choice, the knowledge that nothing about your body is simply given anymore. Everything is decided. Some people want aspects of their existence to be settled, not perpetually open to reconsideration. The stability of biological givenness, even with its limitations, provides a kind of psychological foundation, that unlimited fluidity might erode. Think about transgender individuals currently living with gender dysphoria. Many describe a profound sense of wrongness, a feeling that their body doesn't match their internal sense of self. The technologies we've discussed offer relief from that suffering in ways current medicine can't fully provide. Not just hormones and surgery, but complete biological transformation. Cells that produce the right hormones naturally, reproductive organs that match identity, gametes that allow biological parenthood in the affirmed gender role. The potential for alleviating suffering is enormous. Gender dysphoria causes real pain, depression, anxiety, social isolation, sometimes suicidal ideation. If biotechnology can resolve that distress by making bodies fully align with identities, that's an unambiguous good. We should pursue it, develop it, make it accessible to people who need it. But we should also acknowledge complexity. Some people find meaning in the struggle with their bodies, in learning to live with constraints, in the journey of transition itself rather than just the destination. When transition becomes quick, easy, and reversible, does something get lost? Does the difficulty of current transition paradoxically give it meaning that effortless modification might lack? There's no single answer. Different people will experience this differently. What feels like liberation to one person might feel like loss to another. The key is creating space for multiple responses, not assuming everyone will or should react the same way to these new possibilities. Consider relationships and intimacy. Currently, biological sex plays a role in attraction, partner selection, and sexual dynamics. People have preferences, sometimes flexible and sometimes not, about the sex characteristics of potential partners. What happens when those characteristics become fluid? A person might be attracted to someone who's biologically male today, but chooses to transition to female next year and back to male 5 years later. Does the attraction persist through those changes? Does it depend on current biology, past biology, gender identity, or something else entirely? How do you build long-term relationships when the person you're with might fundamentally change their biology multiple times? These aren't insurmountable problems. People have always navigated change in their partners. Aging, illness, shifts in personality and interest. Biological sex fluidity is just another dimension of change that relationships will need to accommodate. But it's not trivial either. It requires rethinking assumptions about compatibility and commitment that have shaped human relationships for millennia. Then there's the experience of parenthood. Currently, having a child connects you to biological sex in visceral ways. Women experience pregnancy, men don't. That difference shapes parenting roles, family dynamics, even identity. When anyone can gestate, when anyone can produce any type of gamete, those sex-linked experiences become optional rather than determined. A transgender man could choose to carry a pregnancy using his own eggs created through IVG, experiencing biological motherhood despite being socially and psychologically male. A transgender woman could produce eggs and hire a surrogate, or potentially carry the pregnancy herself if she receives a bioprinted uterus. Two men could both contribute genetically to a child and share gestational experience if both have uteruses. The possibilities multiply in ways that completely transform what reproduction means. For some people, this will feel like liberation. Finally, the reproductive role matches the gender identity. For others, it might create new forms of dysphoria. A trans man who wants children but feels distressed by pregnancy's association with femininity, even though he's the one experiencing it. The disconnection between social gender and biological reproduction creates paradoxes that will take time to navigate. There's also the question of what we owe future generations. When we modify our biology in ways that might affect offspring, we're making choices that impact people who can't consent. If epigenetic modifications alter gametes, if bioprinted organs have unforeseen effects on reproduction, if in vitro gametogenesis introduces errors, children born through these technologies might face health problems or developmental issues we didn't anticipate. We have ethical obligations to minimize those risks, to proceed cautiously, to study long-term effects before widespread implementation. But we also have to balance caution against the suffering of people living now who could benefit from these technologies. How much risk to future generations is acceptable to relieve present suffering? There's no formula for answering that. It requires judgment, wisdom, and humility about what we don't know. And there's a deeper question about identity and continuity. If you transition from male to female through complete biological reprogramming, are you the same person you were before? Philosophically, psychologically, legally? If you transition back and forth multiple times, which version is the real you? Or is there no real you, just a series of configurations you move through? These questions touch on ancient philosophical problems about personal identity and what makes someone the same person over time. But they take on new urgency when the changes involve not just beliefs and behaviors, but fundamental biology. We'll need new frameworks for thinking about identity in a world where bodies are radically mutable. What seems clear is that these technologies will create both opportunities and challenges at the most intimate, personal level of human experience. They'll relieve suffering for many people, allowing bodies and identities to align in ways previously impossible. They'll expand human diversity and possibility in beautiful and unexpected ways. They'll give people agency over aspects of their existence that were always previously fixed. But they'll also create new forms of complexity, new questions about meaning and identity, new challenges for relationships and social institutions. They'll require developing wisdom about when to modify and when to accept, how to balance individual autonomy with social concerns, how to navigate choice when everything becomes optional. The human experience of this transformation will be messy, complicated, sometimes contradictory. Some people will embrace it fully, celebrating liberation from biological constraints. Others will approach it cautiously, modifying only what truly matters to them. Still others will resist, choosing permanence in a world of fluidity. All of those responses have validity. What we need is compassion for people suffering with gender dysphoria who need these technologies now, for people uncertain about modification who need time and support to decide, for people uncomfortable with biological fluidity who need social space to maintain traditional patterns. The future of gender isn't one size fits all. It's a landscape of diverse choices, multiple paths, different ways of being human. And ultimately, that's what this entire journey is about. Expanding the ways humans can exist, increasing the options available, removing constraints that have limited possibility for all of human history. Not to force everyone onto one path, but to make multiple paths available, letting each person find their own way toward flourishing. The last generation born with permanent gender represents an ending, yes, but more importantly, it represents a beginning, the start of an era when biological sex is no longer destiny, but possibility, when bodies express identity rather than constraining it, when humans gain unprecedented agency over one of the most fundamental aspects of existence. That future is coming. How we navigate it, with wisdom and compassion and humility about what we don't know, will define not just what technologies we develop, but what kind of people we become in the process. We've explored the technologies and their implications, epigenetic editing, in vitro gametogenesis, bioprinted organs, the social transformations they'll trigger. But there's a question we haven't directly addressed. When does this actually happen? Not in vague terms like the future or eventually, but in concrete timelines that let you understand what's possible in your lifetime, your children's lifetime, your grandchildren's lifetime. Because the answer matters deeply. If these technologies are centuries away, they're interesting speculation, but not personally relevant. If they're decades away, there's something today's young people will encounter in middle age. If they're closer than that, there's something we need to prepare for immediately. So let's be specific about timelines, grounding our discussion in current research trajectories and expert estimates. Start with what already exists. Gender-affirming hormone therapy is available now, widely accessible in many countries, covered by insurance in some contexts. This is baseline technology, first-generation tools that work but have significant limitations. They'll continue improving incrementally, better formulations, fewer side effects, more precise dosing. But the fundamental mechanism stays the same. External hormones overriding your body's natural production. Surgical techniques are also current technology. Vaginoplasty, phalloplasty, facial feminization, breast augmentation, all performed thousands of times yearly with improving outcomes. Again, incremental improvements continue, but we're not expecting revolutionary changes in surgical gender affirmation over the next decade. Better techniques, faster recovery, more natural results, but the same basic approach. Now let's look at emerging technologies. In vitro gametogenesis is furthest along in terms of research progress. We have proof of concept in mice, skin cells converted to functional gametes that produced living offspring. That happened years ago. We have early-stage results in humans. Skin cells converted to cells resembling primordial germ cells, the first step in gamete development. That happened in 2024. The next milestone is completing the full process with human cells, taking human skin cells all the way through to mature, functional eggs or sperm in laboratory culture. Conservative estimates put that 5 to 10 years away. Optimistic researchers suggest it could happen within 3 to 5 years. Let's split the difference and say functional human IVG is likely demonstrated by 2030, possibly earlier. But demonstration in a laboratory is different from clinical availability. After proof of concept comes safety testing, ensuring the gametes are genetically normal, that they can fertilize successfully, that embryos develop properly, that offspring are healthy. That requires years of animal studies and careful human trials. Regulatory approval adds more time. Realistically, even after successful demonstration, IVG probably doesn't become clinically available until the mid to late 2030s. And even then, it will be expensive, available only at specialized fertility clinics, probably restricted to specific medical indications initially. Wider availability, where people can routinely use IVG for reproduction regardless of medical necessity, likely pushes into the 2040s. So if you're young today, in your teens or 20s, you might have access to IVG in middle age. If you're older, probably not in your reproductive years. Epigenetic editing is at an earlier stage. The basic technology exists. CRISPR-Cas9 systems that can add or remove epigenetic marks on specific genes. Researchers use these tools in laboratory settings routinely, but using them in living humans for complex applications like gender transition is much further out. Current epigenetic therapies target single genes for specific diseases. Sickle cell anemia, for instance, where modifying one gene could cure the disease. Even those applications are just entering early human trials now. Expanding to complex traits involving multiple genes across different tissues, like sexual characteristics, is substantially more challenging. Expert consensus suggests therapeutic epigenetic editing might be available for some single-gene diseases by the early 2030s. More complex applications like gender transition would come later. A realistic timeline might be experimental trials beginning in the late 2030s or early 2040s, initial clinical availability in the 2040s, wider adoption in the 2050s. So if you're very young now, children or teenagers, you might have access to epigenetic gender transition in your 30s or 40s. If you're currently in your 20s or 30s, probably not until you're quite old, if at all in your lifetime. If you're middle-aged or older now, this technology likely arrives after your reproductive years have passed. Three-dimensional bioprinting of reproductive organs is the furthest out. We have successful animal experiments, bioprinted ovarian tissue, transplanted into mice, restoring fertility and hormone production. We have early human work with simpler tissues, but full reproductive organs are extraordinarily complex, and we're nowhere near ready for human trials. The timeline here is particularly uncertain. Some researchers optimistically suggest bioprinted organs for transplantation by the 2040s. More conservative estimates push that to the 2050s or beyond. And those estimates are for any bioprinted organs, not necessarily reproductive ones. Given the complexity and sensitivity of reproductive biology, reproductive organs might come later than other applications. A reasonable estimate: experimental bioprinted reproductive organs possibly in the 2040s. Clinical availability for specialized cases in the 2050s. Wider availability unclear, but probably not until the 2060s or later. That means today's children might access this technology in their 40s or 50s. People currently young adults might not see it until they're elderly. People middle-aged or older now almost certainly won't access it. So here's what the timeline looks like in practical terms. Over the next decade, through the mid-2030s, gender transition continues using current technologies with incremental improvements. Hormones get better, surgeries improve, but no fundamental changes in approach. The transgender experience remains roughly what it is today. Powerful medical interventions that significantly change appearance and function, but require ongoing pharmaceutical intervention and don't fully transform reproductive biology. From the mid-2030s through the 2040s, in vitro gametogenesis becomes available, first experimentally, then clinically. This is the first revolutionary change, allowing people to produce gametes that don't match their birth sex, enabling biological parenthood in their affirmed gender role. A transgender woman can be a biological mother. A transgender man can be a biological father. That fundamentally changes reproductive possibilities, even though other aspects of transition remain pharmaceutical. Simultaneously, in the late 2030s and 2040s, early epigenetic therapies enter human trials, initially for genetic diseases, gradually expanding to other applications. Gender transition using epigenetic editing might begin experimentally in the 2040s, offering the first glimpse of transition that reprograms biology rather than overriding it. From the 2050s onward, these advanced technologies mature and become more widely available. Epigenetic transition moves from experimental to established, though probably still expensive and specialized. Bioprinted organs possibly enter clinical use. The combination of IVG, epigenetic editing, and bioprinted organs creates the full suite of options we've discussed. Complete biological transformation, reversible modification, customization of specific traits rather than wholesale transition. By the 2060s and 2070s, these technologies might become routine, at least in wealthy countries with advanced healthcare systems. Gender fluidity at the biological level becomes possible for people with access to the technology. This is when the full social transformation we've discussed really accelerates. So who experiences what? Children born in the 2020s, the supposed last generation with permanent gender, grow up in a world where IVG emerges during their teens and 20s. Epigenetic transition possibly becomes available in their 30s and 40s. Bioprinted organs may be in their 50s and 60s. They straddle two eras, born with fixed biology, but living long enough to see that fixedness become optional. Their children, born in the 2040s and 2050s, grow up taking biological gender fluidity for granted. IVG is established when they reach reproductive age. Epigenetic transition is available, at least for those with resources. Some of them might even receive prenatal interventions that make their biology more flexible from birth. They're the first generation for whom permanent gender is truly optional rather than default. Their grandchildren, born in the 2070s and beyond, live in the fully transformed world we've been describing. Biological sex, as previous generations understood it as a historical curiosity, something their great-grandparents experienced, but they never do. Bodies are customizable. Reproduction is uncoupled from anatomy. Gender is expressed biologically in whatever configuration feels right. That's the trajectory we're on based on current research progress. But there are uncertainties. Technologies could develop faster than expected. Breakthroughs happen. Techniques that seem decades away sometimes arrive suddenly when someone solves a key problem that was blocking progress. Or development could slow. Technical challenges might prove harder than anticipated. Regulatory caution could delay clinical availability. Economic limitations might restrict access even after technologies work. Ethical debates might constrain application. The timeline could stretch out longer than these estimates suggest. And there's geographic variation. These technologies will appear first in countries with advanced biomedical research infrastructure and permissive regulatory environments. Probably the United States, possibly China, maybe South Korea or Japan or parts of Europe. Other countries will lag, some by years, others by decades. Global access to biological gender fluidity will be highly uneven for generations. There's also the question of cost. Early adoption of any new medical technology is expensive. IVG will initially cost tens of thousands of dollars. Epigenetic therapy, probably even more. Bioprinted organs might cost hundreds of thousands. Over time, prices fall as techniques improve and scale increases. But that process takes decades. the full suite of gender fluidity technologies might not be affordable for average people until late in this century. So while we talk about the last generation born with permanent gender, that's really specific to certain demographics. People born in wealthy countries in the 2020s who will reach middle age just as these technologies mature and become accessible. For much of humanity, permanent gender will persist much longer, not because the technology doesn't exist, but because they can't access it. That inequality matters. It creates a world where biological gender fluidity is a privilege of the wealthy and technically advanced, not a universal human possibility. Whether that's acceptable, how we address it, what obligations we have to make these technologies accessible, those are questions without easy answers, but enormous importance. The timeline also affects how we prepare. If these changes are decades away, we have time for measured debate, careful policy development, gradual social adjustment. We can proceed thoughtfully, learning from early applications before wider adoption. But we also need to start the conversations now, because decades arrive faster than we expect. For individuals, the timeline means thinking realistically about what's available in your lifetime. If you're young, you might access IVG and possibly epigenetic transition. If you're older, probably only incremental improvements to current technologies. Plan accordingly. Preserve gametes if you want children, and you're transitioning now, because IVG might not arrive in time for your reproductive years. For society, the timeline means we have perhaps 2 or 3 decades to develop ethical frameworks, regulatory systems, social accommodations for biological gender fluidity before it becomes widespread. That's not much time given how slowly institutions change, but it's what we have. The future is coming. Not instantly, not uniformly, not for everyone simultaneously, but coming nonetheless, with a timeline that makes it relevant to people alive today, not just distant descendants. The last generation born with permanent gender isn't a far future abstraction. They're children in school right now, teenagers figuring out their identities, young adults starting families. They'll live through the transformation we've described, and we're the ones who have to prepare the ground, developing the technologies responsibly, creating the ethical frameworks thoughtfully, building the social structures inclusively. Because the timeline says this is our job, our generation's responsibility. The future depends on the choices we make now.